Hello and welcome to this lesson for higher biology, which today is looking at types of breeding. So what we're intending to do is look at how different methods of breeding are able to produce different results. And essentially we need to explain the difference between inbreeding and crossbreeding, what we mean by inbreeding depression, how it can be avoided and why some plants are less susceptible. We need to be able to explain how a hybrid plant or animal can be created and how this crossbreed can be maintained. And we need to be able to define hybrid vigour and explain why the F2 generation is not as useful. So to begin with, looking at inbreeding. Essentially, during inbreeding we have selected related plants or animals are bred for several generations until the population breeds true for the desired type. So we're taking closely related individuals, siblings, parents and children would be considered closely related. And what we're trying to do is create a true breeding individual. And remember, true breeding is essentially our homozygotes. So homozygous dominant or a homozygous recessive individual would be considered true breeding. And that's because regardless of who they are bred with in the future, they will always pass on one of these particular alleles. Heterozygotes are not true breeding because you can't guarantee which of the alleles they'll pass on. Homozygotes are referred to as true breeding because we can guarantee that they're going to pass that one on. So what we're looking to do in breeding is continually breed related pairs in order to produce lots of homozygote individuals. And ideally, the homozygote individuals will be showing the desired phenotype, the one that we're looking for. The process of inbreeding really works as follows. We take our close relatives and we allow them to mate. The offspring that are produced are going to show some kind of variation. We look over those offspring, and the ones that have desirable characteristics are selected and allowed to mate. However, the ones that aren't showing the characteristics we would like are identified, and we prevent those ones from breeding. We then take the offspring from this section of breeding. Again, they show variation, and the ones we don't want are prevented from breeding. We'll look at the rest that show their desirable characteristics. At this point, we've got a couple of choices though. We can take our superior animals and mate them with others from the earlier generation. So they might not be the same generation. We might be able to take them from up here or up here, one of these lots of offspring, one of the ones that have shown some desirable characteristics. We could also mate them with one another, so just within that generation. Again, those offspring are then going to produce some variation, and we're going to take out the ones we don't want, and the ones that we do want, we're going to repeat that process until it breeds true for whatever trait it is that we're looking for. Now, when we do this, however, what we see is a resulting loss of heterozygosity. So over time, if we imagine that this is our parent genotype, and this is what we would maybe refer to as our F1 and our F2 generation, if we're continually breeding our heterozygotes together, then over time we start losing our heterozygotes, and this can be a bit of an issue. Now, just to go through this quite slowly to give you an idea of how that would happen. If we take our parent generation and say they are all heterozygotes and they have been selfed. Now what that means is the process of self-pollination. If this is our plants, they've been bred using their own pollen has been used to fertilise their own flowers. So those are heterozygotes, and at this point we've got 100% heterozygosity. Now if we were to breed Big A and Little A with another individual with Big A Little A, or it's been bred with itself, so it's got the same genotype, then in our F1 generation what we would expect to see is a homozygote, a heterozygote, another heterozygote, and a homozygous recessive individual. And we would do that through a normal Punnett square. So crossing that with that would result in these 4 genotypes produced. So that's reduced our heterozygosity from 100% down to 50%. Now again, if we're going to take all of the individuals in our F1 generation and cross them with themselves, so they're going to self-pollinate again, So that one is going to cross with the same genotype, this one will cross with the same genotype, and so on. So that would lead us to giving an F2 generation. These individuals would all be homozygous dominant. These individuals, however, would all be homozygous recessive. Our two heterozygote individuals, again though, because they would cross with themselves and pass on a mixture of genotypes, we would have the same variety again. We would have our homozygous dominant, two heterozygotes, and our homozygous recessive, and the same over here. So as a consequence of this, out of all of these individuals, so we've got 16 individuals here, only 4 of them are actually heterozygotes. So we've only got 25% are heterozygotes. If we were to carry on even further, and these individuals were producing four further individuals, this one is breeding with itself and producing these ones, and so on, we would end up with a very similar result. This one producing these ones producing these ones. However, we get to our heterozygotes, and again, our heterozygotes only produce 2 more heterozygotes each, and they will also produce some homozygous individuals, and that's reduced our heterozygosity right down to 12.5%. So over time we can lose this heterozygosity, and that can be a problem. In particular, at this point, if our desired genotype is, say, the homozygous dominant, that's good in some ways. We've got a lot of homozygous dominant individuals. That's working quite well for us. However, we've also got a lot of homozygous recessive individuals, and this recessive allele might start to become a bit of a problem if it's coding for anything that's deleterious, anything that's causing us a problem. And this is what leads to inbreeding depression. So inbreeding can increase the frequency of individuals that are homozygous for these recessive deleterious alleles that are going to cause a problem. And as a result, these individuals will do less well at surviving to reproduce, and that's referred to as inbreeding depression. Now, there are some plants which are naturally self-pollinating. So this means that the pollen they produce will land on the stigma of the same flower, or it might be another flower on the same plant, and fertilise the egg cell. So normally what you'd be looking at is, or in cross-pollinating plants, you'd be looking at the pollen leaving the anther, either blown away or attached to an insect, and being taken away to another flower elsewhere. However, in self-pollination, the pollen literally is just moved from the anther onto the stigma, it grows down its little pollen tube and fertilises the eggs. So that's referred to as a self-pollinating plant, and that's where we get that selfed result that we were just looking at a minute ago with our inbreeding depression. However, there are some plants that are naturally self-pollinating, and as a result, these are less susceptible to inbreeding depression. Natural selection over time has eliminated the deleterious alleles, so those individuals which do wind up homozygous for the recessive alleles essentially just don't survive to reproduce. So over time, we've lost those deleterious alleles, and the ones that are homozygous recessive for any particular allele aren't showing any signs of inbreeding depression. Moving on to crossbreeding. Now, that's a little bit different. That's where, for example, in our plant, we're going to take the pollen of one plant and we're going to put it into the flower of another. We're going to put it onto the stigma of a different plant, often as a different breed. So what we're looking to do is take individuals from different breeds, breeding them together to produce a crossbreed population, and that should have improved characteristics. So, for example, taking a Labrador, taking a Poodle, and breeding these together to produce a Labradoodle would be what we'd be looking to do. And often what we can then do is we would keep these parent populations available to us in order to continually breed our new offspring. This will also work in plants, for example. We might take 2 plants with particular characteristics. Say we're looking at a breed of tomato, a type of tomato that's characteristically quite large. We've also got another one that has low rotting characteristics, and we're going to breed these 2 together in the hope that we produce a large tomato which has low rotting. Again, we might look at it in sunflower plants, where we've got one with a high yield sunflower oil, a high disease resistance, and the offspring therefore have a high yield with sunflower oil and resistance to disease. So these offspring are referred to as hybrids. So they've been produced by crossing two different inbred lines. So over time we've inbred this one to be homozygous, let's say homozygous dominant for the trait that we want. We've inbred this one to be homozygous dominant for the trait that we want. If we continue to inbreed this one, we may see that loss of heterozygosity and inbreeding depression. So what we're doing is we're breeding it with another species, another type of the same species, and we're producing our hybrid offspring. And essentially this gives us a relatively uniform heterozygous crop, and they often have increased vigour and increased yield. And essentially what this would look like is if we were to take our two individuals, and if, for example, we consider that as our first sunflower plant, then the one with the increased oil production, say these two genotypes here, these homozygous dominant ones, are producing our increased oil production. However, this one makes it quite susceptible to disease, this homozygous recessive one here. However, if we're breeding that with the other one, it's got the low oil production. However, it's got good disease resistance. It's got the genotype for disease resistance. If we're going to breed these two together, we get a nicely uniform heterozygous population, which is showing the dominant characteristics for both. We're showing our high oil production, and we're also showing our disease resistance on this side. What we would then do is, rather than breed these ones together, we would maintain these particular crops and keep breeding these ones together to continually produce this uniform set here that's heterozygous and showing both useful traits. The homozygous allele is present, but it's masked by our dominant allele, so it wouldn't show any deleterious effects. So normally we wouldn't breed these ones together. If we do that, we wind up with a huge amount of variation. So our F2 generation would therefore show a huge amount of variation. And all this diagram is, guys, is just a huge Punnett square. But it gives you an idea of just the different types of variation that you might see. Occasionally, you might decide as a breeder to try and breed some of these. You might just be looking for some new varieties. You might just be looking to see what sort of comes up as you breed these two individuals together, to have a wee look and see if you can make any new varieties that may be of use to you. So that takes us to the end of types of breeding. So you should be able to explain that the difference between inbreeding and crossbreeding is that inbreeding is using closely related individuals and breeding them together, whereas crossbreeding is taking individuals from different breeds and putting them together to try and produce improved characteristics. The inbreeding depression is where we have an increase in the number of individuals who are homozygous for recessive deleterious alleles and don't do as well at surviving to reproduce. And some plants are less susceptible to this because they are naturally self-pollinating, and as a result, natural selection has eliminated any deleterious alleles that were present in their genotypes. That a hybrid plant or animal can be created through crossbreeding, taking our individuals from different breeds and breeding them together. And this crossbreed can be maintained by continuing to use the parental genotypes or the parental populations and producing offspring from those. That hybrid vigour, as a result of these crossbreds, are showing increased vigour, increased yields usually, and have a nicely uniform heterozygous genotype. That the F2 generation is not always useful because it shows too much variation as a result of breeding these heterozygotes together.